Hi, my name is Osama Naga. We'll start a new section, Genetic Disorders. You can follow with me in the Pediatric Board Study Guide in the Genetic Disorder chapter from page 83 to 85. My objectives in this presentation to discuss different types of chromosomal abnormalities, indications for genetic tests, the appropriate genetic tests for different genetic disorders, limitations of certain genetic tests. As you know, human body is made of cells, and inside each cell there is a nucleus except the red blood cells. And inside the nucleus there are 46 chromosomes, and each chromosome is packed with coiled DNA carrying a lot of genetic informations. And the chromosomes are numbered 1 to 22 pairs, and they are well known as autosomes or body chromosomes. And number 1 is the largest chromosome, and number 2 is the smallest chromosome. This is how the geneticists they are able to identify which chromosome is which by the size. Number 1 is the largest, number 22 is the smallest, and they are well known as autosomes or body chromosomes. Then the number 23 pair, which is well known as sex chromosomes, and it's XX is for female and XY is for males. And as you see here, if we magnify one chromosome here, you will see this is one sister chromatid and another sister chromatid, and attached with a centromere. And you see here different colors, different staining. This is well known as G banding because of the staining with Giemsa stain. And the concentration is different, more localized or more concentrated in area than others, because the Giemsa stain is specific for phosphate and become more concentrated in the area where it is more adenine and thiamine. That's why you will see different staining as you see here. And this is very important for information about translocations and rearrangement. If we look more closely at one of the chromosomes, you will see there is a short arm and long arm. The short arm marked by P, which is petite or small. So short arm is P and long arm is Q. Very important to understand how the geneticists identify the location of the deletion, for example, or the genetic defect. So usually they identify with a number. So how to understand this number? Exactly like an address, like country, state, city, street, and house number. Very similar. So let's take this example, DiGeorge syndrome. You have here, this is Q, stand for long arm, and the 22 means the number of the chromosome. So the number of the chromosome is the country, which is chromosome 22. Then the state, which is the Q, which is the long arm of chromosome 22. Then the geneticists they divide the long arm of the chromosome to regions or bands. So number 1 is the closest to the centromere, and number 2 is afterward. So as the numbers are increasing, they are more far from the centromere. Same thing with the short arm, it can be divided to 1 and also can be divided to 2. So here there is a region 1 in the short arm of chromosome 22. Then if you expand and look more at this region, at region number 1, you will have more numbers, and it will be numbered the same way exactly. 1 is the closest to the centromere, and 2 is the afterward, and the 3 is the most far from the centromere. So as you see here, 1 here is the belong to the one of the original band. So this is the sub band. So 1 1 1. Then 1 2 3. So you have 1 1 1 2 1 3. So is the correct way to read it is 1 1, not 11. So 1 1 1 2 1 3. Then if you magnify if you take this band and magnify it, you will also be classified to sub band, which is 1 1 belong to this one. So this green belong to the first one, the yellow belong to the second one, the sub band, and the sub band is in red color here is 1 and 2. 1 and 2, 1 because it's close to the centromere, then afterward is 2. So how to understand these numbers? So here the DiGeorge syndrome is a microdeletion in the long arm of chromosome 22, band 1 and the sub band 1 again, and the sub band 2. So this is the how to understand these numbers. So the correct way to read this one, a microdeletion in the long arm of chromosome 22, 11.2. Again, let's take another example. Here you have a 5q. What is 5q stand for? Is the long arm of chromosome 5. This is chromosome 5. This is the long arm of chromosome 5. So 5q means the long arm of chromosome 5. Then another one, if you have this in the sex chromosome, there is no number, you will say X. So XP. XP means the short arm of chromosome X. P stands for petite. So P is the short arm of chromosome X, long arm of chromosome 5. Let's take another example, cystic fibrosis. Cystic fibrosis is an autosomal recessive disease, affects the lung and gastrointestinal tract, usually associated with chronic lung disease and failure to thrive and malabsorption. So the scientists were able to identify the genetic defect in cases of cystic fibrosis in 1989. So they give it this number. This is the address on the chromosome. So how to read that? This is the long arm of chromosome 7. This is chromosome 7. This is the long arm of chromosome 7. And the defect is localized in band 3. And here is the band 3 and subband 1 and subband 2. So how to read this correctly is a genetic defect where in the long arm of chromosome 7 3.1.2 because of defect in the cystic fibrosis transmembrane conductance regulator, which is responsible for chloride ions. Chromosomal abnormalities, aneuploidy, and structural abnormalities. This is the main chromosomal abnormalities. Aneuploidy means abnormal number of chromosome. For example, 47 instead of 46. Structural abnormalities, the numbers are the same, but there is a problem with the structure, like for example, translocation, duplication, and deletion. So this is structural abnormalities. Genomic imprinting will explain that. Mitochondrial inheritance, expansion of trinucleotide repeats. Let's start with the abnormal chromosome number or aneuploidy. First example of abnormal chromosome number or aneuploidy is monosomy. Monosomy means the loss of one chromosome, so 45. So 45 instead of 46, this is Turner syndrome. As you see here, just one X instead of 2. So one X instead of 2, this is Turner syndrome. The second most famous example of aneuploidy or abnormal chromosomal number is trisomy, which is gain of one chromosome. So you have 47 instead of 46. So you have 47, 47, 47, 47 because of gaining one extra chromosome. If this location is 21, chromosome 21, you have one extra here in chromosome 21. This is well known as Down syndrome. If you have one extra chromosome in the chromosome 18, so this will be Edwards syndrome. If you have it in 13, you will have, this is Patau syndrome. If you have it in the sex chromosome and additional X here, this will be well known as Klinefelter syndrome. Structural abnormalities or aberrations is deletions, duplications, translocations. Deletions means a part of the chromosome, like here, part of the chromosome or a sequence of DNA lost during DNA replication. So you see here the genetic material here, or part of the chromosome is missing here in this area. This is called deletions. Or the opposite, the part of a chromosome or DNA sequence is duplicated during the process of DNA replication. And the translocation will explain in the next slide. Translocations is exchange between the chromosomes. We have reciprocal translocations and Robertsonian translocation. Reciprocal translocation can occur between any of the chromosomes. For example, if we take 1 and 2, if you have one piece of chromosome 1 goes to 2 and one of chromosome 2 goes to 1, if they are the same, this is balanced translocation. Many people are healthy living with balanced translocation. If it's too much of 1 goes to 2 or too much of 2 goes to 1, this will be well known as unbalanced translocation, and this can be associated with health problem. Robertsonian translocation is same idea, but the translocation occurs between specific chromosomes, well known as acrocentric chromosomes. Chromosomes with very short arm and normal long arm. These chromosomes are 13, 14, 15, 21, and 22. When 14 combined with 21 will give you a longer chromosome combination of 21 and 14. And of course, this will be negligible, so this patient will have 45 chromosome and is a carrier. This patient is healthy, no problem, as long as there is no children. But once they have children, the problem can happen. If they pass 21 copy to another 2 copies of 21, they will have trisomy 21, and this can be a cause of Down syndrome, Robertsonian translocation. If they pass, for example, one copy of 14 to the other two copies of 14 will have trisomy 14. This can cause miscarriage. If it's 13, will be Patau syndrome. So this is very important to understand the idea of Robertsonian translocation, because this can be associated with Down syndrome. So again, Robertsonian translocation occurs in the acrocentric chromosome, chromosome 13, 14, 15, 21, and 22. Let's start a new section, Mendelian inheritance. This was derived by Gregor Mendel. Is a monk from Austria. He planted peas in the back of the church at 1856 and 1866. Then he tabulated a Punnett square, and he recognized the organism. It has genes, and the genes it has alleles. If the alleles are similar, is well known as homozygous allele. If they are different, it is heterozygous alleles. Autosomal dominant. Autosomal means body chromosomes, means not sex chromosomes. This means that any genetic condition associated with autosomal can affect both males and females equally. Dominant means that the affected allele can be expressed by itself. Like in this case, for example, let's pretend this father is having Marfan syndrome, and the affected allele will be in uppercase because it's dominant. So any child is having or carrying the affected allele, the uppercase M, will be affected with the Marfan syndrome. So if we use it here, the Punnett square, this is for the dad, this is for the mom, you will have a 50% chance of having a child with Marfan syndrome, another 50% chance to have a child without Marfan syndrome. So every time the mom is pregnant, if the mom is healthy, of course, she has a chance of having an affected child, 50%, and also she has a chance of having a child without Marfan syndrome, 50%. So every time, regardless how many healthy children she has, regardless how many affected, she may have all of them with Marfan, she may have no child with Marfan, she may have 50 and 50. So this depending on the time of the mom is pregnant. So every time she's pregnant, she has a 50% chance to have Marfan or without Marfan. So again, autosomal dominant presence of one abnormal gene on one of the autosomes, chromosome 1 to 22. Both sexes are equally affected. Both sexes can transmit to offspring. No generation is skipped unless not completely expressed. 50% chance to have a child with the disease. Every affected child has a parent with the disorder, except the new or spontaneous mutation. Examples of autosomal dominant: osteogenesis imperfecta, neurofibromatosis, polycystic kidney disease, and achondroplasia. Autosomal recessive. Autosomal means body chromosomes, means the conditions associated with autosomal can affect both males and females equally. Recessive means that the affected gene or allele require another one in order to express the disease. Different than autosomal dominant, one affected allele will express the disease. Here you require two together. So in this case, for example, this is a father with sickle cell trait, this is a mother with sickle cell trait. If they decide to have children, there is four possibilities. 25% chance to have a healthy child, 50% chance to have a child carrying the disease or having sickle cell trait, and another 25% chance to have a child with sickle cell anemia. So the autosomal recessive, they can have carrier. Autosomal dominant, there is no carrier, either sick or not sick. Here the chances are 25% to have a child with sickle cell anemia, 25% chance to have a child healthy is not a carrier, is not having a sickle cell anemia or anything, just healthy, and 50% chance to have a child carrying the disease. Autosomal recessive involves mutation in both copies alleles at a gene locus, non-sex chromosomes or autosomes 1 to 22. Both sexes are equally affected because it's autosomal. Males and females can each transmit a copy of mutated gene. Recurrence risk for parents with a previous affected child is 25%. The risk of parents who are carrying a mutated gene to have an affected child is 25%. Consanguinity increases the risk of having an offspring with an autosomal recessive disorder. Example: cystic fibrosis, thalassemia, sickle cell anemia, familial Mediterranean fever. X-linked recessive is inheritance of mutated gene on X chromosome or sex chromosome X. So this has to do with the gender. Only females can transmit the disease to their sons. Boys are affected, and female carriers are generally unaffected. Female carriers, they have 25% risk of having an affected son sick with the disease, and 25% risk for a carrier daughter, and 50% chance to have a child not affected at all, that is not inheriting or carrying the mutated X-linked gene. Examples of that: hemophilia A and B, Duchenne muscular dystrophy, and G6PD deficiency. X-linked dominant, the dominant gene is carried on the X chromosome, affects both males and females. 50% chance of having an affected child if one parent is affected. Diseases associated with X-linked dominant are very rare. Examples: X-linked hypophosphatemic rickets, incontinentia pigmenti, Aicardi syndrome. Aicardi syndrome is agenesis of corpus callosum or dysgenesis infantile spasm and chorioretinal lacuna or holes. Rett syndrome, Albert syndrome. Albert syndrome is deafness and hematuria and renal failure. Most of the cases of Albert syndrome are X-linked dominant. Genomic imprinting is very interesting. If the mutated gene is transmitted from the father, will give you a different disease than if it's transmitted from the mother, the same mutated gene. Like in this example, we have Prader-Willi syndrome. The affected gene is transmitted from the father. The affected gene is located in band 1113 on the long arm of chromosome 15. The inherited allele from the mother will be silent or inactive. Prader-Willi syndrome is well known with hypotonia after birth and failure to thrive initially. Then they start to have hyperphagia, obesity, small hands and feet, and hypogonadism and intellectual disability. In case of Angelman syndrome, is the opposite. The affected gene is transmitted from the mother, and this gene is the same gene of Prader-Willi, but is transmitted from the mother, which is a mutation on the band 1113 on the long arm of chromosome 15. The inherited allele from the father in this case is silent or inactive, like is not there. The Angelman syndrome, the presentation, they are happy personality, frequent laughter, intellectual disability, speech delay, microcephaly, epilepsy, and ataxia. Remember, Angelman is maternal. Let's look at this pedigree and try to understand. The yellow is the affected or the diseased person. This mom here is diseased or affected with a certain condition, and here is the father here is affected with certain condition. But none of the generation of this father is having a disease, but all the generation of the mother, they are having the same disease. This is a classic pedigree of mitochondrial inheritance. The mother is the one who will be able to transmit the disease to all her generations, but the father will not be able to transfer the disease to his generation because of lack of mitochondrial substance in the sperm. The mother, she has in her eggs lots of mitochondrial substance and genes, and this will be transmitted to all her generations. Mitochondrial disorders are transmitted mainly from the mother, is unipaternal inheritance from the mother only. Why? Because the egg, it has from 100,000 up to 1 million mitochondrial DNA, while the sperm, it has very few, so it's rarely the sperm or the father will be able to pass abnormal mitochondrial DNA to offspring, but the mother will be able to pass the mutated or abnormal mitochondrial DNA to all of her offspring, and this will cause unipaternal inheritance of mitochondrial diseases. Mitochondrial diseases like MELAS. MELAS is mitochondrial encephalopathy, lactic acidosis and stroke, and Kearney-Sayre syndrome, which is progressive ophthalmoplegia and cardiac defect. There is another form of inheritance, is multifactorial inheritance. It means that there are many factors, for example, genetic and environmental factors. Often one gender is affected more than other. For example, like in cases of DDH or developmental dysplasia of the hip is more common in females than males. The question we need to ask ourselves before we order a genetic test: why does my patient need genetic testing? Indications for genetic testing: previous family history, consanguinity, multiple miscarriage, death in utero, congenital anomalies, developmental abnormalities like autism, neurologic disorders like epilepsy, multi-organ dysfunction. Congenital anomalies, you have either minor anomalies or major anomalies. If you have more than or equal 3 minor anomalies, it's worth it to do the genetic testing, like ear tags, 2 to 3 tooth syndactyly, epicanthal falls, hypertelorism, or one major anomaly is enough to do the genetic testing, like brain anomalies, congenital heart defect, cleft lip, cleft palate, omphalocele, and limb absence. Chromosomal analysis, the fastest and easiest, is a blood test, which is peripheral blood lymphocytes, testing the lymphocytes or the white blood cells. If a newborn in the nursery, for example, received packed red blood cell transfusion, this does not affect the genetic testing because we tested the white blood cells and not the red blood cells. Other sources like amniotic fluid or chorionic villus sampling, cultured skin fibroblast, bone marrow cells. Karyotype is a test to identify and evaluate the size, shape, and number of chromosomes. This test is very beneficial. This is the most common test, karyotype, to be used to diagnose trisomies like Down syndrome, sex chromosome anomalies like Turner syndrome, Klinefelter syndrome. Visible deletions and duplications and translocations easily identified with karyotype. But we need to know the limitation. The limitation of karyotype, that a G-band blood karyotype can never exclude extremely subtle chromosome abnormalities. In this case, microarray will be beneficial, but no need to do microarray for a classic presentation of Down syndrome or Turner or Klinefelter. So this case is easily identified with karyotype, not microarray. DNA microarray is able to measure the level of gene expression of all genes within a cell. It is useful for detecting micro deletions, micro duplications, and specific breakpoints, which cannot be detected with regular karyotype. Examples or indications for using DNA microarray: multiple congenital anomalies, autism, and developmental delay. FISH test is an excellent choice to order if you suspect a syndrome associated with microdeletion, for example, DiGeorge syndrome. If you do karyotype for DiGeorge syndrome will look normal, but FISH test will be able to detect the microdeletion. How? By detecting a specific DNA sequence in a chromosome by using DNA probe containing fluorescence labeled probe, and this will detect a specific sequence on the chromosome. If it gives two signal, this is normal, like here giving two signal, and if it gives just one signal, this is abnormal. And this is the long arm of chromosome 22, it's very short, and this is a microdeletion on the long arm of chromosome 22, which is DiGeorge syndrome. Also can be used for Williams and other syndromes associated with microdeletion. If this test is negative and you still highly suspect DiGeorge syndrome, microarray will be the best choice. FISH test is widely available, is an excellent test to order for cases suspected with microdeletion, but very important to know the limitation. It is possible that rare, very small deletions may not be detected by FISH test. In this case, microarray will be very helpful. This is the end of this presentation. Thank you. This video is only for educational purpose and not intended to direct care of any specific patient. Please consult your physician or the physician of your child for the correct diagnosis and the proper treatment. None of my videos can be superior or an alternative for the opinion of an experienced and licensed healthcare professional. Thank you again.